Welcome back, students. This is Module 2.1.4, Why are cells small? And in order to understand this concept, you have to understand first that the volume of a cell is proportional to the needs of a cell. So the more volume it has, the more cytoplasm, the more stuff, and it needs more oxygen, more food, more waste removal to meet its needs. On the other hand, cell surface area is proportional to the ability of a cell to meet its needs. And this is because the surface area is where the membrane is, and membrane is what allows needed things to come in and needed things to go out. And so the more surface area, the more transport, and the better able to meet needs that a cell is. Now, often, biologists and scientists put together something called a surface area to volume ratio, or simply surface to volume ratio. And this relates the cell volume to the cell surface area. And there's some calculations down here you can look at in a moment, but I want you to understand that as a cell gets bigger, its needs increase, but they increase by the cube as the volume increases, if you look in the units, but the surface area only increases by the square. And so that means, in layman's terms, that the volume increases faster than the surface area. And this has some serious implications. Okay, the volume increases faster than surface area. Therefore, the cell needs, which are proportional to cell volume, will outstrip the ability to meet the cell needs, which, of course, that ability to meet needs is proportional to transport or to surface area. And so cells are often small because of surface to volume constraints. This is an important thing that you should be able to explain. And you should look at these calculations here. It starts out with a cell that has a diameter of 0.5 centimeter, and you can see the surface area is 0.79 centimeters squared, and the volume is 0.06 centimeters cubed. Not that the numbers matter that much. Just get down to the ratio, surface to volume ratio. It's 13.17 to 1. Now, if we double the diameter of the cell, and you can see the corresponding surface area and volume, but let's just get to the ratio. Look at the surface to volume ratio. It has it is less than half, 6.04 to 1. And we can see, again, if you increase the diameter of the cell and to 1.5, and now it's the surface to volume ratio becomes 3.99 to 1. And so what we're seeing is as the cell gets bigger, the surface area to volume ratio becomes less and less favorable. In other words, the needs are greater than the ability to meet the needs, or they're becoming greater. And at some point, you'll have a precipitous failure where as a cell gets too big, it will lose the ability to survive. It won't be able to meet its needs. So this is one important explanation as to why cells are small. And you should invoke this. If I ask you this on an exam, you absolutely should talk about surface area to volume relationship. So here is a summary. Why are cells small? Because of surface volume constraints explained previously. That's the major reason, and it better be in your answer. But there are other reasons why cells are small. Having many small cells allows greater compartmentalization, and you can have greater specialization. You can also have the ability to isolate disease cells, and you can lose disease cells, no problem. A one-celled organism that loses one cell is in big trouble. But if you have 1 trillion cells and lose even 1% of your cells, no big deal, and that's millions and millions of cells. A third reason why cells are small is that many small cells rather than one large cell means cell death is not organism death. I kind of already discussed that. Now, this concept of surface area to volume extends beyond the explanation as to why cells are small. It is a very broad concept with many applications in biology. So as I say here, an important concept with broad application in living things beyond why cells are small. One example, respiratory structures. For example, it's been said that human lungs have enough area to wallpaper a handball court. Now, I don't know if that's true or not, but it is true that the lungs have very many small alveoli, these little compartments, and they have a tremendous amount of surface area. And so this is, again, where surface area is being maximized because the function of the lungs is to provide exchange or transport, if you will. A second application of surface area to volume is in thermoregulation. For example, heat budget. If you're very, very cold, then you'll put your arms and roll up in a ball, put your arms in and roll up in a ball and decrease your surface area, reduce the temperature exchange with a cold environment. You can also consider the house that the Inuit live in. You might call the Inuit Eskimos. It's an igloo, and that has the lowest surface to volume ratio to minimize exchange with the frigid Arctic environment. Can't get any better than half a sphere in terms of minimizing your surface area. And we can see in physiology and biology that surface area is often maximized when a surface is used for production or absorption. Think of your cerebral cortex. Here over on the left, this is a mammalian brain, and all these wrinkles provides greater surface area, and that's because in the cerebral cortex, that's where if you have more neurons, you can have more complex interactions, and your brain is capable of higher order functioning. Think of microvilli in the intestine. Microvilli are little finger-like extensions of the cell membrane. So instead of being a smooth membrane, the part of the cells that line the intestine have these finger-like microvilli. This greatly increases the surface area. This last picture shows the underneath of a mushroom, and it shows gills radiating out from the central stem, and all these gills provide great surface area, and it's on the edges of these gills that spores are produced. And so you should consider surface area to volume when you are beyond the context of why cells are small. It has, as I say, very broad application. And here is a scanning electron micrograph, a transmission electron micrograph of microvilli. And this is what absorbs food in your intestines. And it just greatly increases the surface area. So you can see down here I've written food. That's where food will be passed along. And all these finger-like things are extending out. And this has been sliced in preparing the slide. But these microvilli would be 360 degrees around the tube where the food is going. So I hope you can appreciate that. I want you to notice at the end of this lecture, and this is the end of the lecture, the last module about cells, we have a little bit of a quiz, and so you can practice with cell structure here. Of course, the answers are on the next slide, but I recommend that you are able to know the basic structures that I've given you here in this lecture. So thank you for your attention.